Hello everyone. First of all, I would like to thank the organizers for the opportunity to present our latest research on intrinsic functional connectivity in patients after hemispherotomy. Our study group at the University Hospital Bonn consists of 28 patients, 17 of which are female, and we collected data from immediately after surgery until 20 years post-surgery. We have even two interesting cases in this cohort where we collected the MRI data before surgery and at multiple time points after surgery. When we had a look at the structural data of those patients, we found that depending on the surgical technique that was applied, the amount of gray matter that was intact on the isolated hemisphere varies a lot. So we decided to split up our group further into those patients where we could only analyze the connected hemispheres, but also those patients where we could analyze one connected hemisphere and one isolated hemisphere because there was enough vital gray matter tissue left intact in the brain. Our image processing stream was as follows. We first of all drew manual lesion masks on the T1 weighted images and performed a parcellation into 7 networks that was further divided into 200 parcels, which is a very common parcellation. We discarded all parcels where more than 50% of our voxels were inside the lesion masks, and further, if in any network more than 50% of all parcels were discarded, we would only analyze the connected hemispheres. But if in contrast, for all networks, less than half of all parcels had to be discarded, we analyzed both the connected and isolated hemisphere, which was the case in 9 patients. Moreover, we had a 12-minute resting state fMRI scan of all patients at 3 Tesla, which we processed through fMRI prep, incorporating the manual lesion masks, and finally performed our connectivity analysis with NiLearn. We performed our analysis in 3 steps, basically, that I will guide you through now. First of all, we proved the parcel homogeneity to show that this parcellation into 7 networks and 200 parcels is even applicable after hemispherotomy. Next, we quantified the connectivity between parcels in comparison to controls and also longitudinally over time. And as a third step, we quantified network efficiency in comparison to controls and also over time. And basically, we will come to our conclusions that we draw from this. So let's start with our first step to prove parcel homogeneity. To show that the connectivity profiles in every parcel are homogeneous, we technically calculated 3 types of correlations. First of all, the within parcel correlation, which is the average correlation between every voxel and the parcel that it belongs to. Next, the inside network correlation is the average correlation between every voxel and the average time course of parcels that lie in the same network, so inside this network. And third, the average correlation between every voxel and all parcels that lie outside, so in a different network. In healthy controls, one would expect that the correlation within the same parcel is obviously highest, followed by the inside network correlation and least the outside network correlation. So I've drawn this on a graph for all parcels, so approximately or a maximum of 200 parcels per hemisphere. And also on this glance, we see that within seems to be greater than inside or outside in controls, but as well on the connected and isolated hemisphere. To make this even clearer, we calculated the difference between within correlation and inside network correlation and averaged this across all hemispheres and see that this is greater than 0 for the isolated and connected hemispheres. And last not least, also the difference inside minus outside network correlation is bigger than 0 on the isolated and connected hemispheres of a hemispherotomy, which allows us to conclude that this pattern within greater inside, greater outside network correlation is intact on the connected and also on the isolated hemisphere after hemispherotomy, and further that this parcellation into 200 parcels and 7 networks is applicable after surgery. Now that we have shown that the connectivity profiles in every parcel are homogeneous and that this parcellation is applicable, we move on to quantifying the connectivity in the isolated and connected hemispheres. To quantify connectivity, we calculated the correlations between the average time courses between all pairs of parcels. So if we now first of all look at those connections where both parcels lie in the same network, we see that this is slightly reduced on the connected hemisphere after surgery and even more reduced on the isolated hemisphere after surgery. Now those between network connections where both parcels are in different networks, we see that this is increased on the connected hemisphere and even more on the isolated hemisphere after surgery. Now, if we look at those patterns on network level, we see that especially those correlations or those connections are increased after surgery, especially in the isolated hemisphere, as drawn in blue in this graph, where networks that are counted as unimodal networks, like the visual network or the somotor network, are connected with higher order, so transmodal networks, like especially default mode network or control network. So now let's look at the connectivity profiles longitudinally with a mixed effects model. We found that within network connections increase over time, both on the connected and isolated hemisphere, with a stronger increase on the isolated hemisphere across all networks. If we look at the slopes individually per networks, we see that a stronger increase on the isolated hemisphere is especially present in transmodal networks, while on unimodal networks there is less increase on the isolated hemisphere. More interesting again between network connections, here also we see an increase in connectivity both on the connected and even stronger on the isolated hemispheres. If we look at this per network, we see a gradient showing that the strongest increase in connectivity on the isolated hemisphere is found in the transmodal networks, so in the default mode network and control networks, followed by limbic and salience networks, while there is the least strongest increase in the unimodal networks, visual and somotor network. Finally, as a last step, we looked at the network efficiency and quantified it in comparison to healthy controls and also longitudinally over time. At first, we analyzed global efficiency as a measure of network integration. Here we found that global efficiency was significantly reduced as compared to controls on the isolated hemisphere, while it remained identical on the connected hemisphere. In contrast, local efficiency, which is a measure of network segregation, here we found a pattern of slightly reduced local efficiency on the connected hemisphere and even more reduced local efficiency on the isolated hemisphere almost across all networks, while this was especially significant in the default mode network. If we look again at the changes over time, we see that global efficiency increases significantly on the isolated hemisphere, while it remains almost stable on the connected hemisphere. Next, I brought data from 2 patients where we collected functional data before and after surgery. Here we see that in patient A, which are the top 2 lines, the global efficiency drops almost by half after surgery, but then recovers very quickly to almost the value as it was before surgery within the first 6 months. Patient B is a little bit different. Also here, after surgery, the global efficiency, which is even lower than in patient A before surgery, drops a little, but only on the connected hemisphere. It recovers and also not as fast as in patient A. If we look again at the slopes with regard to local efficiency, we see that there is a stronger increase again on the isolated hemisphere than on the connected hemisphere. And here is the same pattern as with regard to between network connectivity. This effect is especially strong in transmodal networks, while the changes over time in unimodal networks that have very specialized tasks are not as strong as in the higher cognitive function transmodal networks. Now, as we reach the end of my talk, please let me summarize the conclusions that we draw from our data. First of all, we have shown that the very common parcellation into 7 networks and 200 parcels is applicable in the connected and isolated hemisphere after hemispherotomy. Next, we've shown that there is a functional reorganization which is seen as an increase in global and local efficiency and an increase in connectivity between networks in both hemispheres after hemispherotomy, while this is stronger on the isolated than on the connected hemisphere. And third, this remodeling is even stronger in transmodal networks, so those networks that mediate higher cognitive functions than in unimodal networks. And more as an outlook, we see this as a necessary, even though not a sufficient condition for an island of awareness that is given in the isolated hemisphere. By that, I would like to thank my lab, the Translational Neuroimaging Group in Bonn, all collaborators on the project, especially my supervisors, Theodor Rüber, Professor Surges. I would like to thank you for listening, and I'm happy to receive questions.